I'm Gemma Mendonça, and I have been at Seagate Technology working in their research group for the past 6 years. Oops, now this one is like too sensitive. There we go, okay. And I hail from the United States, the lovely state of Minnesota. My 8-hour flight from Minnesota arrived here at about 7:30 this morning, so it's been a very long time since I slept. If my words are not as clear as one might hope, I hope you'll be sympathetic to my situation. Seagate is mostly known for their hard drives. That's their main product. They make and sell a lot of them. But in the research group, one of our roles is to explore alternative data storage media. Wow, I'm not touching it. I'm just like thinking about it. It must be on a timer. So one of the alternative storage technologies we are exploring is DNA data storage. And I'm really excited to share some of the experimental results we have from our chemistry lab, where we've been developing and testing new and unique chemistry methods to make writing DNA more practical. I don't think I really need to spend much time on this slide. We all like DNA data storage. It's dense, you don't need to plug it in. If you treat it right, it's stable, it's easy to make copies of, and as we're gonna hear, I think in some of the other sessions, it's got some really interesting compute capabilities. The less fun aspect of DNA data storage is why it's not currently mainstream. I think most of us are aware of the many challenges that DNA data storage faces. I've highlighted just some of the main ones that we focus on for the data writing portion of things. We know that writing data in DNA takes a long time. It's currently too expensive to be practical. One of the other problems that I want to highlight, just because I haven't heard people talk about it as much, and I'd love to have some conversations with others while I'm here, is about the volumes of liquid reagents that are consumed when the DNA is being written. We all know DNA is really dense. So if you can write a terabyte and it's this tiny little speck, or this tiny little pellet or something. But what did you use to make that? You probably used a large volume of liquid reagents, and if you're using liters or tens of liters or hundreds of liters per terabyte, that's not going to be practical at the petabyte, exabyte scales. We have been working on some liquid handling solutions to help with that. I can't remember which one is the laser. To help with this problem of reducing the volume of liquid reagents we're consuming. We have that work published recently in Lab on a Chip. I'm not going to have time to talk about it much today during our time together. What I'm going to be talking about is more the chemistry aspect of things. Unique write chemistry that we've been developing and testing to make DNA writing both faster and cheaper. We have an article coming out really soon in Nucleic Acids Research. I'm very excited. It's been accepted. It hasn't been published yet. I think it's going to come out in the next couple of weeks. So I'm going to give you a bunch of spoilers. You don't have to read it. I still encourage you to read it. I'm really proud of it. I think it's really cool. First, I'm going to focus on the write architecture that we've been developing. So instead of writing the DNA nucleotide by nucleotide, we wanted to use an assembly-based approach. We think it's promising for making DNA write faster. I think it's fairly straightforward to envision. It's more likely that DNA write will be faster if you're putting together many nucleotides together at one time rather than nucleotide by nucleotide. So we wanted to focus on an assembly-based method. Our method is built on a foundation of 2 libraries of DNA. The first library contains data-encoding oligos. We call them symbols, and their job is to just encode the data. We write the DNA by assembling the symbols in the desired order to encode the desired information. we have to tell the symbols which order they should be assembled in, and we do that using our other library of DNA oligos called linkers, because they link the symbols together. So if we have symbols that we're trying to assemble together, first we need to attach one linker on either side of each symbol. So here's an example of a symbol and linker complex. This colored region here is the symbol. There is a left linker and a right linker attached to it. So once we have those complexes, we can assemble them together in an assembly reaction using another type of oligo called a splint. So this splint oligo hybridizes to the complementary linker ends, and then you add a DNA ligase, and it chemically links these DNA strands together at these junctions so that they're no longer 5 separate pieces, but they form one continuous strand. In this example, we call it a construct. So you can imagine putting 5 symbols together into a construct. Where this method starts to be powerful for potentially speeding up the write process is when you can do that assembly step again. So imagine you've made a 5-symbol construct. What if you do that 5 times? So you have 5 5-symbol constructs, and then you do another assembly step where you put those 5 constructs together. Now you're left with a DNA strand that contains 25 symbols rather than just 5, but you only did 2 reaction steps. It gets even more exciting if you can do that a 3rd time, where you put together 5 25-symbol constructs. Now you have a DNA strand that contains 125 symbols. We also have an advantage where if we're assembling prefabricated DNA pieces, we can buy them in bulk. If you buy DNA in bulk, as with anything, it's much cheaper than if you buy it in really, really minuscule quantities. So when you purchase or synthesize oligos in bulk, it makes them pretty cheap. It makes them the cheap component of this reaction. But when you do a DNA assembly reaction, you introduce a new cost, and that is the cost of the ligase. Ligase is the protein, again, that chemically links these together. Proteins tend to be expensive, and in this case, they're by far the most expensive component of the reaction. So we got to thinking, what's cheaper than a protein? Another strand of DNA is cheaper than a protein. So DNA does not only encode information, which is what we like to talk about. There are certain unique sequences of DNA that can also perform a function. Instead of folding up into a double strand, double helix, excuse me, they fold up into a unique structure, sort of like a protein. And that, there's another laser up there. This unique structure, as you can see in this example, endows this short DNA sequence with a function. So we looked through the literature to see if there was a DNAzyme that could ligate 2 DNA strands together. Lo and behold, we found this DNAzyme here. It's called E47. So E47 is a DNA strand, and I'm not doing that. And it has this catalytic region here where I didn't do that either. I think there's a ghost. So here in this looped region, that's the catalytic core of the DNAzyme. And coming up from the top here and off to the side here are 2 binding arms. Those 2 binding arms hybridize to 2 separate DNA strands. They call them the S2 subunit, and the S1 subunit goes up here. And when you add zinc as a cofactor, the DNAzyme will ligate these 2 DNA strands together at this junction to form one continuous DNA strand. In order for the ligation to take place, the S1 strand has to go through an activation step first. So you do have to have a specific active group at the end of the S1 subunit. For the S2 subunit, you can just have the 5' OH, which is standard whenever you order an oligo or synthesize an oligo yourself. It's usually going to have a 5' OH group. But if on the 3' end of the other strand, it has to have a phosphate. And you have to further activate this phosphate by adding EDC, which is a carbodiamide, and an imidazole. So that just forms a more reactive intermediate that the DNAzyme can work with. So it's a two-step process. First, you activate the phosphate, and then you perform the ligation with the DNAzyme. So since DNAzymes are going to end up being cheaper than proteins in the long run, nobody had ever assembled multiple pieces of DNA using this technology. So we wanted to test that in the lab and see if that would work. And if we apply this DNAzyme assembly method to the architecture that we talked about with using linker-directed symbol assembly, it's going to look something like this. So in the first set of experiments, we wanted to take a symbol, which is this red thing here, and we wanted to attach two linkers to it. So one, the left linker and the right linker, and then these are the two DNAzymes that we used to do that. So this is a 3-piece reaction, and when we were setting up the experiments, we said, let's try to make a 5-symbol assembly. So we did this 5 times to make 5 unique pieces. And then in the second step, we wanted to do another assembly where we joined those 5 pieces together using 4 DNAzymes and 5 pieces to do a 5-piece assembly. And we wanted to check and make sure that we were actually ordering the symbols in the way that we wanted to. We started with our experimental tests with this first reaction here, where we joined 2 linkers to each symbol to make these 3-piece assemblies. I really wish Lisa would stop doing that. So we used a technique called gel electrophoresis to analyze the results of things. Nope. Is it like I can move it there? Anyway, it's all right. So we use gel electrophoresis to run the results of the ligation reaction. And for those of you a lot of you probably know what gel electrophoresis is, but some of you might not. So gel electrophoresis is a technique used to separate DNA strands based on their size. So you load the DNA strands into the gel matrix and you force them to migrate through the gel matrix. Smaller DNA strands are going to migrate faster through the gel matrix than the large strands. And the strands strands of a similar size tend to migrate together. For example, here in this band of DNA, that's 30 nucleotides long. This next band up here is a little larger, 40 nucleotides, 50, 60, etc. So we wanted to make sure that we made the right 3-piece assembly. We have in this first lane here our size marker, and this is just something we had commercially synthesized. We didn't assemble this piece. This is just what we expected to find. And then in the second lane here, we have our crude reaction. So you can see a band right in line with our size marker. So we can tell that, yes, we made a 3-piece assembly like we expected. We also see some other bands, and I can explain what those are. So this band here, at about 57 nucleotides, is the catalytic strand, so the DNAzyme that we use to do the assembly. Down here, we have some unreacted subunits, and then up here, we have some 2-piece assemblies. So a ligation took place, but it was only 2 pieces rather than the expected 3 pieces. So we can see that, yes, we got a lot of the expected product, but we also can see that the yield is not 100%. So we still see lots of truncated and assemblies and unused subunits. So we do a purification step. I used HPLC or gel electrophoresis to purify the three-piece assemblies from the gel matrix. And then in the second assembly step, in the first assembly step, we made 5 symbol linker complexes, and then we performed a second assembly step to join those pieces together. So we did the same thing. We used gel electrophoresis to analyze those results on a gel, and then we can see, again, this is our size marker, and then this is our expected band. And the band is there, but it's pretty faint. So we did get the expected product. It wasn't as high yield as I was hoping for. So that's okay, it's a proof of concept. So we purified that full-length product, and then we PCR amplified it and then prepared it for sequencing so we can check and make sure that the symbols were assembled in the order we actually designated. I think I'm going to skip this slide. If you're interested in my PCR problems, you can ask me later. It's hard to do PCR on DNA strands with a lot of repeats, but we figured out how to get that better. Then after we performed the PCR amplification, we did DNA sequencing. So you can see the Sanger sequencing traces on the side here. So we use that to confirm that, yes, the symbols we expected to see were in the order that we expected. So we assembled the correct product, and we were able to see that really clearly with the Sanger sequencing. We also did Oxford nanopore sequencing to check and see if there were any variants. So we compared the sequences that we got with Oxford nanopore with a reference sequence, and we wanted to see, hey, are there any variants? Are there any mutations? And we did not find any. So that really indicated pretty clearly that we had made the sequences we desired, and we did this 3 times. So we made 3 different 5-symbol complexes, and we rearranged the symbols to show that, yes, we can direct the order of the symbol assembly using these DNAzymes. And as I mentioned, it's in press, our article is in press, so keep an eye out for it if you're interested. And thank you to my teammates back home who helped out with this work a lot. Contact me and of course, come speak to me if you're interested in learning more about this. I'll also touch on some of the other work we're doing. It's also really interesting. So we're working on, I mentioned this in the beginning, lab on a chip solution to both automate the chemistry and to, in the future, help scale down the reaction volumes that we need to make those DNA assemblies. So we have this publication in Lab on a Chip. We were working with a digital microfluidic platform. For those of you who aren't familiar with that, it's a liquid handling system where you have basically an array of electrodes, and you coat them with a hydrophobic coating. And then if you put a droplet on one of the electrodes, you can move the droplet from one electrode to the next one simply by actuating the electrodes. By turning them on and off, you can move it around these different electrodes. So we were able to build a proof of concept one, and then the hope or the plan is to be able to expand this. So this is a tiny little board. It's just to show the chemistry demonstration, but we hope to, if for DNA data storage scales, you would need many, many electrodes, hundreds of thousands, maybe millions electrodes on your array, and you would have many droplets moving around all at the same time. So you would need to, we call it the traffic problem. How do you make sure each droplet moves where it needs to go without crashing into anything else or without getting stuck and caught up in gridlock? So we were working with the University of Minnesota. They helped to develop this algorithm, and they have some published results there too. For future work, we're continuing to work on the DNA chemistry. So the DNAzyme chemistry, we proved that it can work to assemble the DNA pieces in the order that we want. Certainly is at the proof of concept phase. Haven't gotten to the point yet where we could perform multiple sequential assembly steps to give us that geometric growth of the DNA strand. In order to achieve that, the assembly step would have to be higher yield, particularly as we add more pieces. So the efficiency dropped pretty dramatically the more pieces we added in the assembly reaction. So we would need the assembly reactions to be higher yield, and they would also ideally need to be faster. I wasn't optimizing them for speed yet, but we're going to certainly need to do that in the future. And also, to make it more automatable, we would have to simplify some of the steps, particularly purification of the full-length product. If you remember the gel image that I showed, the reactions were still full of truncated assemblies and unreacted subunits. So we needed to get rid of those pieces, and we need to find a more automatable way to do that. And that, if we can accomplish those things, then we'll be in a good place to start to perform the multiple tiers of assembly. I apologize for the technical difficulties, but that was the material I had for today. I'm happy to take any questions.
Thank you very much. Do you hear me? Yes. Thank you very much, Gemma, for this challenging talk. So, any question, comments? For Gemma?
Yes, thank you for this wonderful talk. From my experience, DNAzymes have a much lower activity compared to protein counterparts. Could you comment on this? What's the percentage? Or yeah.
Sure. They are typically a bit slower than proteins. Some of them are faster than others. This one's not too bad for speed. I know some of the fastest ligating DNAs, the fastest DNAzymes that are kind of close to protein speed, I think, are the RNA cleaving ones. I don't think the reaction rate is going to be the limiting step, at least not yet. I think it's going to be more of like figuring out the purification steps. If, let's say you're doing a ligation with a T4 ligase in the lab, it takes, 20 minutes, 30 minutes. That's how long it takes for a DNAzyme reaction too. So it's going to be similar time scales. It'll be a little slower. I'm more concerned about the yield than I am about the reaction time, particularly for the multiple step assemblies, or the multiple piece assemblies, excuse me.
Yes, and another question here, another there.
Hi, thank you for the talk. Just wondering, when you do the PCR, how you manage to target the full-length assembly, because you did Sanger after all, so afterwards. So I guess you managed to have a homogeneous full-length assembly.
Mm-hmm. Yeah, for purifying those constructs, I used page purification of page gel. So I was, cutting out the bands. It's very labor intensive. It worked for the paper, but it would not work on the scales you need for DNA data storage.
Okay, cool, thanks.
Another question?
My question is, how did you design your oligos, the positional and the different kinds? Did you use any?
Very carefully. It was a pain. So I just use an oligo analyzer software to make sure that those ends were not going to interact with each other too much. In the future, I've seen some papers, developing algorithms to avoid collisions between DNA strands, specifically for the purposes of DNA data storage. So that would certainly make things easier in the future to design like a large system. You would need software aids to do that, but I think that's doable.
Thank you. Okay.
Hi, thank you so much for your talk. It was really interesting. I wonder if you tested different lengths of sequences with your DNAzymes and if you saw different results with the different lengths and efficiencies?
Sorry, is the question, does the length of the subunits affect the
exactly.
activity? My colleague did test that to some extent. I know Mengdi looked at some of the shorter lengths. I think they were, between, and if the lengths were in the couple of tens of nucleotides, like I think she did maybe 20 to 60 or something. There wasn't that much of a difference, but the ones I was using are much longer than that. So when you're making a construct is almost 600 nucleotides long, I do wonder if that played a role. I haven't tested it. So, good question.
So we have one last question here.
Thank you very much for the talk. My question related to the amplification steps, because you didn't cover that, and I think I briefly saw something about a specific enzyme, because even for the rather short compared to probably your targets that you're currently synthesizing, you probably already have quite a lot of repeats, and I was wondering how you were handling that.
Sure, yeah. That's an easy one because I had the material in there. I wonder if I can go back to it since you asked about it. Can I go back? That's okay. I could tell you. So when you try to PCR amplify DNA with a lot of repeats in it, you tend to see this laddering effect where you expect only one band, but for some reason you see a lot of bands. And I dug into that, and apparently it is a known phenomenon when you're amplifying DNA containing repeats. Just because I guess if it has repeats, it tends to form secondary structures, and then as the DNA polymerase bumps into those secondary structures, it slips. They call it slippage. I don't think anybody really knows what that means. But what we found that really worked for us was to use a particular DNA polymerase, and it had a strand displacement quality, property, so that if it encountered some sort of secondary structure, it would kind of unzip it. We tested a few different kinds of strand displacing DNA polymerases, and one worked better than the other. So we found the one that worked the best for us was this deep vent, and it had the exonuclease activity removed. We tested this other strand displacing polymerase called SD, strand displacing, but we still saw lots of laddering with that. So we moved forward with this DNA polymerase.
Thank you very much again, Gemma.